You know, I love the summertime. The leaves are green, the birds are chirping, and the weather's warm. God, I hate cold weather, you know? I hate winter, it's just the worst, you know? I wish there was a way that I could close my eyes in the fall and then just wake up at springtime. Skip winter altogether. Surely that's impossible. Right?
Now the food has disappeared.
Their energy needs plummet.
Why do animals hibernate?
Figuring out how to get humans to hibernate could be a life-saving therapy.
Cold has forced most animals to burn more energy,
Its body temperature drops.
and all life processes slow to a minimum.
I want you to imagine if we humans could hibernate, what would the world then look like?
Now, while this isn't a reality for us humans, animals, and in our case mammals, can go into hibernation to protect their bodies from harsh environmental conditions, like the cold of winter. Animals go into periods of what we call torpor, which are basically long slumbers. These periods of torpor can last from days to weeks, and in between these periods are waking hours. We call this euthermia. It is in these periods of torpor that animals' metabolic rates drop to as little as 5% their normal rate in order to conserve as much energy as possible. In fact, their ambient body temperatures drop from the normal 37 degrees Celsius to 0 to 5 degrees Celsius. And in ground squirrels, the normal heart rate is 200 to 300 beats per minute, but in hibernation, the heartbeat drops from 3 to 5 beats per minute. All in all, hibernation can last up to 6 to 7 months. And in this time, animals can conserve up to 90% of their energy. Wow, that's pretty crazy. I mean, how is that all done? Well, we're going to look at a couple of ways. First, it starts in autumn with a process called hyperphagia. Here, animals who go through hibernation basically binge eat a whole bunch of lipids. They increase their body mass by 50% just from fat. Well, we call it adipose tissue. And there are two classifications of adipose tissue, and they're varied by their color. We have white adipose tissue and brown adipose tissue. We're going to look at the brown adipose tissue because it has to do with thermoregulation, or basically maintaining body heat. This is crucial during hibernation. Now, since we have all these fat reserves that were built up during hyperphagia, we need a way to access these in order to get their energy. We do this through fatty acid binding proteins, or FABPs for short. Now, lipids aren't soluble in water, which means they're also not soluble in the bloodstream. They have to bind to these FABPs in order to be transported through the body in the blood to the cells that require their energy. Luckily, when in hibernation, animals can upregulate the transcription of this fatty acid binding protein gene through the gamma isoform of the transcription factor peroxisome proliferator activated receptor, or PPAR gamma for short. In general, nuclear receptors are proteins that can bind and sense certain molecules. Retinoid X receptor is a nuclear receptor that binds to PPAR gamma. This heterodimer, which just means two proteins working together as one complex, can then bind to specific promoter regions in the DNA of the target genes that produce the FABPs. This increase in transcription means there are more FABPs that can carry lipids around the body, which will provide more energy for harvest. When the lipids reach the cells, they are able to diffuse across the membrane. But now we have to turn them into energy, and for that to happen, we need to convert them to acetyl-CoA. Its goal is to enter the mitochondrion in order for it to enter the Krebs cycle. Through a ligase enzyme, the fatty acids are able to bond with a free coenzyme A to produce a fatty acyl-CoA. This acyl-CoA can now be shuttled into the mitochondrial membrane on a mitochondrial enzyme called carnitine palmitoyltransferase 2. Now that it's in the mitochondrion, beta oxidation cuts the acyl-CoA into two carbon fragments that link up with the coenzyme A to produce acetyl-CoA. Now that we have our acetyl-CoA, it goes through the familiar Krebs cycle and the electron transport chain in order to produce ATP for energy. This is how animals in hibernation get most of their energy: fat metabolism. You can say we bypass glycolysis and pyruvate oxidation. So if fat metabolism is the main energy source, why is it that carbohydrates such as glucose aren't going through glycolysis and pyruvate oxidation? Initially, glucose does go through glycolysis, producing two pyruvate molecules. Now normally these two molecules should go through pyruvate oxidation and be catalyzed by an enzyme called pyruvate dehydrogenase. During hibernation, Currently unclear transcription factors increase the production of an enzyme called pyruvate dehydrogenase kinase. Now this does what the name suggests. It takes pyruvate dehydrogenase and it phosphorylates it, essentially deactivating it. This means that the enzyme that normally does the pyruvate oxidation is no longer functioning. This means the molecules of pyruvate aren't able to go through pyruvate oxidation. We're stopping the flow from glycolysis to pyruvate oxidation. We'll look at the implications of this shortly. Much of the hibernating animal's potential energy is stored in triglyceride molecules. Now we've already established that the fatty acid chains are able to go through the Krebs cycle to produce energy. But what about the glyceride backbone? It turns out that the glycerol backbone of the triglyceride can be turned into G3P. This is the molecule found at the end of glycolysis's energy investment phase. This means you are able to continue glycolysis until you produce one pyruvate molecule. In fact, the process of converting this G3P molecule into pyruvate is expedited by the upregulation of certain glycolytic enzymes during hibernation. For example, PGK1 catalyzes 1,3-biphosphoglycerate to 3-phosphoglycerate, and ENO1 catalyzes 2-phosphoglycerate to produce PEP. Both of these enzymatic increases actually speed up the rate of pyruvate production. But now we're stuck with pyruvate that is unable to go through pyruvate oxidation. What do? Instead, pyruvate undertakes anaerobic respiration. Pyruvate is turned into lactate while oxidizing NADH to NAD+. Luckily, gene transcription for the enzyme that enables this process, which is LDHA, is increased during hibernation, meaning a faster rate of anaerobic respiration is allowed. From there, the newly formed lactate molecule enters what we call the Cori cycle. It is transported to the liver and is converted back to pyruvate. Additionally, one NAD+ is reduced to NADH. The newly formed pyruvate then meets another pyruvate in the liver and enters gluconeogenesis. This is just a fancy term which means building back up a glucose molecule. Our new glucose molecule then travels from the liver to a muscle and goes through glycogenesis to be added on to a glycogen strand for even more energy storage. Now let's talk a little more about gene transcription in hibernation. Gene transcription in hibernation is very, very important. Essentially, the body needs to service what is kind of an alien state. This is totally different from a normal regulating body. The organism must benefit from maximum energy efficiency, and it must go about different processes in different ways. In fact, studies have shown that in ground squirrel hibernation, there is a 45 to 55% decrease in ribosomal subunit function. This means that translation as a whole is decreased during hibernation. Let's look at a really cool example of hibernation gene expression. Hypoxia-inducible transcription factors, or HIF1, respond to hibernation. These transcription factors feature oxygen-sensitive alpha subunits. In hibernation, breathing and heart rate slow, so HIF can sense this decrease in oxygen. Now, HIF1 is known to upregulate a variety of genes that is known to enhance hypoxia tolerance and better deliver oxygen to the tissue. For example, the gene coding for vascular endothelial growth factor, or VEGF, is a signal protein that stimulates the formation of blood vessels by bonding to receptors of the endothelial cells. These are the cells that line the interior of blood vessels. This helps the organism deal with decreased blood flow or apneic breathing, as it delivers more oxygenated blood to more tissue. Finally, let's take a look at how hibernating organisms stay warm enough to function. This is called thermoregulation. The metabolism of glucose to create ATP is only about 50% effective. The rest of the energy is lost in exergonic reactions. Due to this fact, hibernating organisms have found it beneficial to, when they need to, generate a whole bunch of ATP for no real net benefit other than generating more heat to keep them warm. One of thermogenesis' most fascinating aspects is its ability to uncouple oxidative phosphorylation from the electron transport chain. What this means is that the ETC functions as normal, with 6 protons being pumped into the intermembrane space. But unlike normal, these protons do not turn ATP synthase as they move back down to the matrix. Instead, another protein, specifically an uncoupling protein, or UCP1, that goes by the name thermogenin, creates another channel into the matrix that the protons are able to travel through. Normally, thermogenin's channel is closed. it is inhibited by the purine nucleotides ADP and GDP. Thermogenin can be activated in two ways. One, a short-chain fatty acid allows thermogenin to become a symport, allowing both a proton and the short-chain fatty acid entry into the matrix. Or two, a long-chain fatty acid acts as an allosteric activator, binding to thermogenin to allow the passage of a singular proton into the matrix. Here we see the allosteric inhibition caused by the nucleotides is overcome by increasing the amount of the allosteric activator. So how does thermogenin get produced? Well, when the body is exposed to cold, just like hibernating bodies are when they hibernate, the body senses that stimulus and releases the hormone norepinephrine from the ventromedial hypothalamic nucleus, or VMH for short. It's located around the center of the hypothalamus and is responsible for heat regulation. The norepinephrine is an amino acid-derived hormone, so it must travel through the bloodstream and bind to a cell receptor protein, as it cannot diffuse across the membrane. Only lipid-based hormones can do that. The specific receptor protein that norepinephrine binds to is beta-adrenergic receptor, which releases an intracellular signal transmitted by cyclic AMP and protein kinase A, leading to the breakdown of triglycerides. That does two things. It frees fatty acids for the Krebs cycle to produce energy, and it enables fatty acids to act as the allosteric activators for thermogenin. As we said, this allows the protons to move back into the matrix without turning ATP synthase and wasting that heat energy. The norepinephrine activated protein kinase A also phosphorylates a transcription factor called CREB. This transcription factor induces the expression of the gene that is responsible for thermogenin production. So when the cold triggers norepinephrine to be released, it actually causes the production of thermogenin. However, CREB also induces expression of ICER, which is a competitive transcription factor to CREB. ICER acts as a repressor on sites that the CREB would normally bind to. This explains why, when an animal experiences longer periods of neuroepinephrine release, the amount of thermogenin mRNA produced is not as much as we would expect. Indeed, the CREB transcription factor actually produces ICER transcription factors, which in itself stops thermogenin from being produced. As soon as the production of thermogenin is triggered by the CREB, it is inhibited by a repressor that was also triggered by CREB. This would appear to cause a problem, but it's solved because the amount of thermogenin produced before ICER binded to its repressor site is satisfactory for animals to keep their body heat. Evidently, there are many methods that organisms use to conserve energy, maintain metabolism, and generate heat during dormancy. It's remarkable how truly strange the concept of hibernation is when you really think about it. The systems of an animal are being thrown for a loop, having to essentially rework numerous processes all in order to prey upon as few energy resources as possible. It is astounding how just sheer nature itself has developed these methods of creating and coping with these immense metabolic changes. And that's what makes hibernation truly awesome. Thanks for watching.